You're listening to Science Coast with Mallory Havens and
Chris White.
And today our guest is
Sarah Powers.
Welcome back.
So today we're going to talk about Dr. Powers' research lab. And this past year, she was the recipient of a Brother Rapp Award here at Lewis University, which provides Dr. Powers with time and money in order for her to pursue her scholarship. So, Dr. Powers, can you tell us a little bit about your scholarship?
Sure. My background training is with a focus on understanding the immune system, but more specifically, I am interested in understanding the way in which we make our immune cells. So there is a need in order to generate these immune cells from stem cells that live in your bone marrow throughout your entire lifetime. It's a very well studied, characterized, stepwise process because all of these cells in our body have the same genetic code, right? So we have the same fundamental DNA, but what makes a skin cell versus an immune cell is really deciding which genes are being used in each of those. And along the way to make our immune cells, not all immune cells are exactly the same, and they are going to have different functions. So there needs to be decisions that are made on a kind of lineage basis to know what cohort of genes are being used in order to make them.
So I'm wondering, when you say make an immune cell, in my head, I'm imagining that immune cells are all very similar, and then what you're maybe is customization of an immune cell, something similar to what you're talking about?
I would say that customization is a good descriptor, as well as the fact that the cells are not really ready, prepared, or licensed to do their work on day one.
So they're generic.
So we start off with a stem cell that is capable of making really all of our blood cells. So this is red blood cells, right, which are not immune cells themselves, but I kind of want my red blood cells around, right?
Oh,
They're
yeah.
really important.
Only if you want to get oxygen around your body.
It's kind of a big deal. We also rely on our blood stem cells to make things like platelets. Also important if you want to
not bleed to death
and have clotting. And then we have a variety of different cells that are important for fighting off disease, right? And to protect us against pathogens. And they don't all play the exact same role. They're complementary as a network or a team approach in order to go ahead and get rid of whatever that threat is. But in order to make that diversity of the response, they each have to go through their own developmental program to get there.
So how do you study immune cells and the making of immune cells and the customization of immune cells?
Well, in my research group, we are not trying to do all of it at once, right? Because that's a little bit overwhelming. My background research, I got started in trying to understand how we make B cells or B lymphocytes. So they're some of those cells that are important for making the memory response that's important for vaccines. But they're a little bit temperamental to just go ahead and have those cells grow in a culture setting like we have in our research labs here at Lewis University. So what my current research group is focused on is trying to understand how neutrophils are made. So neutrophils are part of what we call the innate immune response. They are probably some of the most important responders that are going to get called to the site of infection as soon as you're exposed. They're going to be short-lived, but masterfully powerful at destroying pathogens. So they can take things up from outside of themselves. They can gobble up infected cells. They can even go ahead and plug holes. So there's some really great videos out there looking at neutrophils infiltrating into a mouse ear after it's been bitten by a sandfly with an infection, and they just zoom in and they go ahead and they plug that gap in order to make sure that nothing else gets in.
Almost like first responders.
Yeah, that's how I teach it in class, is say that they're like the paramedics. So when your immunological memory is your adaptive immune system, and it only gets to meet pathogens if they get past the innate immune system. So you have the first responders on the scene. If they can take care of the problem before it spreads, then it's over with. But if they can't and they have to call in the physicians and the nurses and whatever, that's your adaptive immune system that learns and remembers. Whereas neutrophils, they don't care what kind of pathogen you are. They will fight any foreign thing.
How do they know whether something is foreign?
That's a complicated question. But there are certain things that we don't have as humans naturally. So if we see it, we know that's foreign, like double-stranded RNA. We make single-stranded as humans. Or there are certain proteins that we don't express, or like flagella shouldn't be floating around in our bodies. But Sarah can expand on that, I'm sure.
Yeah, and there's, similar factors that are present in other pathogens, but molecularly they're different. So certain bacteria will use sugars that are composed fundamentally differently than our sugars are made. And so the kind of probes that can detect these things can decipher between us and something else.
It's really at the molecular level.
Yep.
It is.
And that's what's really fascinating to me. I know very little about biology, but if I were to get into biology, it's that molecular biology that seems so interesting.
Why do you think that we are focused on molecular things?
Because you're awesome.
Plus, our own cells have essentially ID tags or licenses that say, hey, I'm part of your body and I'm not foreign, that our immune cells can kind of check, go around and survey and be like, let me see your ID, which is why when you get like an organ transplant, they might have an ID, but you're like, is that a fake ID? And that's why you give immunosuppressants. So the immune system is like, I can't really tell. I don't have my glasses on. So I guess I'll let you stay.
I'm getting the impression that it's slightly more complicated than that.
Yes, it is. It is. But just to give you, that is like the intro level of what I say in Gen Bio 1 to be like a basic idea of what's happening. Then we dive a little deeper.
Okay, so what do you hope to achieve with the research that you're doing?
So we're trying to understand how my favorite protein, which is called cyclin D3, has its hands in a couple of different cellular processes. So, cyclin D3 has been understood for decades now as an important factor for helping cells decide when it's appropriate to divide from one cell into two. So they integrate information from outside of the cell that's saying, grow now, it's appropriate. And that's one of those factors that helps the cell transition to the point where it starts copying its DNA so that ultimately we can have division and each of the resultant cells has a full genome, right? And don't get me wrong, it's important for these cells to divide because if we need to make neutrophils in order to respond in the context of an infection, we get the numbers we need because they are dividing. But some of my doctoral research focused on learning that cyclin D3 also has its hand in the pot of regulating what genes are being expressed in a cell. So it's dual function. We just don't quite understand yet the mechanism for how that works.
So when you bring up replication of cells, the first thing that pops in my head is cancer, because cancer is also a replication process gone wrong.
Mm-hmm.
So am I just off base here?
No.
No, you're actually really on base, right? So one of the avenues that my research students have taken to try and understand parts about the cyclin D3 protein that might be altered in certain circumstances is they went through databases and looked at different versions of cyclin D3 that are present in certain cancers. So it is known to correspond with cells that are proliferating abnormally. And so we've gone and we've found those changes in the genetic code in these cancer samples. And we're using that as part of our investigation in order to say, well, maybe if you have these alterations in the protein, it changes the fundamental role, and that could help us dissect how it's working in a normal circumstance.
Do you think that the cyclins can actually function as transcription factors? And just for clarity for people listening, those are proteins that go and allow genes to be expressed, so they promote the making of RNA from the DNA.
So we think that cyclin D3 is working indirectly.
Okay.
We think that it is somehow binding to or changing the ability of other transcription factors to do their work.
Okay.
So we don't have evidence that it binds directly. And we've tried to see that, and that doesn't seem to play out. But there are certain other transcription factors that are patterns that associate with genes that we know that cyclin D3 seems to modulate. It's also not crazy to think about cyclin D3 doing something with regulating DNA, because its kind of sister protein, cyclin D1, is really well characterized for dealing with DNA damage and fixing that part. So these molecules make it into the nucleus and they're interacting with the DNA and doing something. We're just still trying to figure out exactly how cyclin D3 is doing its cool stuff.
So it does go into the nucleus?
For sure.
Okay.
Yes, and I've seen that ad nauseum under a microscope. So it likes to glom up, right?
Yeah.
And it likes to stick. From my graduate lab, we also have some really cool fluorescence in situ fish experiments in order to go ahead and we can see that cyclin D3 sticks itself adjacent to RNA polymerase.
Oh, okay.
We just don't know how exactly things are binding together.
Okay, that makes sense. RNA polymerase is the enzyme that makes RNA from DNA. It makes the polymer RNA. Thus, RNA polymerase.
It ends in ASE, so it's an enzyme.
Oh my gosh.
Okay, we didn't practice that.
No, that was cool. So for those of us, including myself, who are like, okay,
sorry,
I hear the words
we
that
nerded
you're
out
saying.
for a
No,
second.
but I loved it. It was absolutely fantastic. But okay, what does that mean? So when the research is done, how is this going to benefit people? What's going to be possible because of all of this knowledge that you're generating?
So you have to understand basic science before you're able to approach it in different disease states, right? So if you fundamentally don't know how something works, you don't understand what goes wrong in different diseases. So establishing like normal baseline healthy how stuff works then allows you to see in certain diseases what is different, and then you can try to get back to normal. But until you know what normal is.
So the research, is it possible that it will lead to drug development? Is it, therapeutic? Is it just getting a better understanding of how the biology works, and then we're not sure what the outcome is going to be?
So I would say first and foremost, we want to understand the biology, right? I would argue the importance of this protein because it is an intersection between two kind of fundamentally perhaps conflicting cell processes, right? A cell is figuring out how to do its day-to-day function when it's not dividing, and that is maybe the gene expression regulatory role, but then it also is helping these cells know when they should be dividing. If we have alterations to the protein that maybe favor one or the other, that could be informative in states where we have an abnormal version of cyclin D3 in cancer. If we want to be able to have good, effective, personalized medicine, then we need to understand the whole of how that molecule is working, right? What if we go ahead and come up with a strategic approach to use personalized medicine, but we're doing it in the wrong way, right? So maybe we're flipping these cells from having a cyclin D3 that is functioning normally for the cell proliferation state, but is abnormal at the gene regulatory state. And that's what's making the cancer happen. And if we don't treat it the right way, are we causing the other problem? Does that make any sense?
Right, it totally makes sense. Does it make sense to you, Chris?
Yes. I'm just going to nod my head. No, but what I hear you saying is that you're contributing to the development of basic understanding of biological function, which others who will follow you will use and incorporate into their research and build upon what you've done. Mm-hmm. And that I understand because that's what I did when I was an active researcher as well. And it was I would refer to my work as being incredibly esoteric, where there wasn't any obvious application or benefit, but we do it with the understanding that the benefit is going to happen in the future. As we develop knowledge, an example was like with radio waves. When radio waves were discovered in the 1800s, it was like, okay, what do we? I mean, now we understand what these are and how and what the physics behind them, but nobody had any idea how incredibly revolutionary they would be in the 20th century.
Right.
And I feel like what you're doing is similar. You're developing this knowledge, and we don't fully understand all of the implications of how it's going to benefit people in the future.
Right.
I think we kind of divide scientific research into basic science, where it's just trying to understand the scientific process, like what is actually happening, and then translational, which is trying to take that basic science into a therapeutic. So when you hear the phrase like bench to bedside, that is the whole process of basic science through drug development to a patient, which is much more extensive. So translational medicine is like, we know the fundamentals, now we're working on the therapeutic, and basic science is just trying to understand the process.
So Dr. Powers, what else would you like us to know about what you're doing in your lab?
So I have, in addition to the neutrophil project, we're branching out into, I have some students that are getting up and running with C. elegans worms. And so we're starting to think about how the immune response works in these worms, because it's way different than ours.
Why C. elegans?
Well, they're kind of cool. I think Dr. Havens would go ahead and...
Yeah, they're incredibly well understood model organism. We know exactly how many cells they have. We know exactly, you can trace the cell from when it goes from a single cell egg to the adult version. We know the process for all 954 cells that they have. So it's very well understood. You can control them in a lot of different ways, and they're very easy to treat with different reagents or chemicals or therapeutics to change how they behave, because you can basically feed them things and it will go into all their cells, which is not how it works for people. But it's a great model organism that we can use to study higher order things by just breaking it down into something a little bit simpler.
Which, in the context then of worms and the immune response, it's very interesting to me that the pathways that are important for protecting them against pathogen are like directly overlaid with the aging process.
Mm-hmm.
So there are a lot of neat things that are there. I also have some interest in kind of evolutionary theory just in general, but in the context of the immune response, we can think about C. elegans worms are total oddballs, right? Like they have gone down their own evolutionary pathway compared to even other invertebrate organisms, such as Drosophila melanogaster, the fruit fly. So there's remarkable conservation of things for the immune response in the fruit fly that is present in us. C. elegans worms doesn't have a lot of those fundamentals. They've gone on their own branch of the tree of life, and they seem to have lost a lot of those mechanisms. So I don't know. I think it's interesting, and the students are really excited to get working on it, and they have their own lines of investigation they're proposing. So cool stuff.
Yeah, for sure. You've been listening to Science Coast. The views, thoughts, and opinions expressed are the speaker's own and do not represent the views, thoughts, and opinions of Lewis University. The material and information presented here is for general information purposes only. The Lewis University name and all forms and abbreviations are the property of its owner, and its use does not imply endorsement of or opposition to any specific organization, product, or service. This podcast was produced in the WLRA podcast studios at Lewis University. Visit lewisu.edu for more information about Lewis University. I'm Mallory Havens, and I'm with
Chris White.
And our guest is
Sarah Powers.
Thanks for listening.
Bye.